Hello, sixth grade science students, once again. Today we're on lesson 10. Our question is, what do cells need to grow and make more of themselves? The objective for today is to explain evidence that shows that cells need food to grow and make more of themselves. We're going to watch an investigation and analyze the data to answer that question. What do cells need to grow and make more of themselves? And how does that relate to healing? We're going to use bacteria cells. First, we'll compare these to human cells. On the left, we have two different types of bacteria. And on the right, we have human skin, human bone, and human muscle cells. These are all on high magnification under a microscope. What's similar and what is different? They are cells. They are microscopic. The bacteria are individual living things. The bacteria cells are each a living individual, but the skin, the bone, and the muscle are all cells that make up tissue, that make up organs, that make up a much larger organism or individual living thing, like a person. So each bacteria is an individual living organism or a separate living thing. In larger plants and animals, many different types of cells work together to make one living organism. Scientists like to work with bacteria because they are really simple. They're just made of one cell, and each cell can carry out all the functions they need by themselves. In contrast, animals, which are made of lots of cells, are much more complex and need lots of different types of cells that work together to perform very specific functions. So, sometimes, studying simpler organisms that are made of cells can give us some insight into what might be happening in our own cells. We are going to try to get evidence to convince us that bacteria cells need food and nutrients to grow and make more cells. Scientists use E. coli bacteria, that's the species name, E. coli, because they are easy to use in the lab. They are non-disease causing versions or strains of them, which means there are also some that can make you sick, but we're not using those. And they share similar features with other cells. We want to test or investigate if bacteria will make more cells when they have more food. So we need a way to give the bacteria food, but in a way where we can control the amount they get and that it's all the same kind of food or nutrients to make it a fair test. And then we will only change one thing. We call that the independent variable. We'll only change the amount of food that we give to the bacteria. How will we do that? We'll use this stuff called agar. Agar is a gel on which to grow cells in a lab. And we can do it in different ways. But it's a dry powder made from red algae, commonly found throughout the world's oceans. As a powder, it dissolves in boiling water. When an agar solution cools, it forms a semi-solid jelly. And scientists create agar plates by pouring hot agar solution into a petri dish, or these shallow glass dishes with a glass cover, and letting it cool down. And then we can put different amounts of nutrients into this. When we dissolve it in the water, some of it can be very high nutrition, some of it can be in a form that there's no food there for the bacteria. So preparing agar plates for growing bacteria. Here's how they do it. In this video, we see them pouring an agar plate. They've got the hot liquid here with the powder dissolved in it. They've got the little petri dish. They shake it up to make sure it's all dissolved. And he wears a glove because it's hot, remember. They just had boiling water put into the powder. And then he pours it out into the dish. or I should say the person pours it out into the dish. They cool the plate, and after it's cooled, it's set up like a firm jelly. Pretty cool. Let's see, properties. This is what it looks like after it's cooled off. This is the properties of the agar when it's cold or cooled out. And they're going to show that to us right there. Look at that. It's set up just like Jell-O in your refrigerator. They can actually pick it up. He's pulling it up there with that little spatula. And you can see it's flexible, it's soft, and it all holds together. Pretty cool stuff. So, how will bacteria respond? How will bacteria respond to different nutrient levels? In this investigation, step 1, we prepare solutions of agar with different levels of nutrients. We've got some with no nutrients up to pure nutrients. So it's going to be 0% nutrients, 25% nutrients, 50%, 75, and 100. Which one do you think will have the most growth of bacteria? Will it be the one with no nutrients or food, or the one with all nutrients, or somewhere in between? So, are they going to streak the plates? What does that mean, to streak the plates? Here's a video showing us how they take E. coli bacteria and they put it onto these agar plates, which don't have any bacteria growing on them because they were boiling, so they would have killed off anything in there. So they take the plates. Let's see. They've got the cooled plates there. And I see there's a fire. He's heating his metal hoop up so there's no bacteria growing on it. And he touches it down to the agar so it's cool enough that it doesn't kill the new bacteria from the E. coli solution that they want to put on that plate. So the only bacteria there will be the one he's scraping on right now. See how he just scrapes back and forth, and he gets his bacteria. There he's cleaning off his hoop again in the fire. And he says the tool streaks down the plate. And now what's he doing? He's creating a culture of the bacteria on there. Oh, I see. This is the different plates. He's doing it in all the different plates with the different amounts of nutrients. That's what he's doing. Okay. So then they put them in an incubator, which keeps it at a certain temperature, the best temperature for growing the bacteria, for 2 days. And after 2 days, what do you think is going to happen? What will happen on the plate with no nutrients, and what will happen on the plate with the most nutrients? Let's think about that. There's pictures of the plates. 0 nutrients, 25% has some, 50% it's got a streak there, the 75% it's got a bunch, and the 100 has even more. So what does that tell us about what do bacteria need to grow? Well, each of these little white fuzzy things on there is a bacteria colony, millions and millions of bacterias. So the white fuzzy stuff is bacteria, should say colonies. That says coles. Should say colonies growing on the agar. It looks like having more nutrients in the agar resulted in more bacteria colonies growing. So what are we going to do with that information? You're going to draw and write on a slide on the next item in Canvas that will let you uh, create an argument or say why that evidence shows that cells need foods to grow and to multiply, to increase in numbers.